Good afternoon, everyone. Welcome to today's EMSL Learn webinar on stable isotope labeling and metabolic visualization at spatial scales. The EMSL Learn webinar series is designed to provide information to the scientific community on resources available at EMSL for research in the biological, environmental, and computing sciences. Our webinars are posted on the EMSL Learn section of the EMSL website, so we invite you to visit that section to see past webinars. If you want to stay informed on upcoming webinars and other upcoming proposal calls, please sign up to receive our emails from EMSL or follow EMSL on social media, LinkedIn, X, Instagram, Facebook. The links will be posted in the chat. Today, our presenters include Sophie Lehman, a PNNL Earth scientist, and Vimal Balasubramanian. I hope I got that right, Vimal, an EMSL biologist. They will discuss the capabilities available through EMSL's Stable Isotope Laboratory and provide more information about stable isotope probing. Jeremy Bogor, an EMSL chemist, is going to chat with you about EMSL's NanoSims capability, and Dusan Vilishkovich is going to walk you through a new EMSL capability available through MALDI for untargeted molecular imaging of roots and rhizosphere. The instrumentation discussed today is available to the scientific community through our open ficus call. Letters of intent to submit a ficus call are due on March 6th. There will be time at the end of today's webinar for questions, so we encourage you to post your questions in the Zoom chat so that our speakers can answer them during the Q&A section. And so with that, we are going to begin with Sophie.
Thanks everyone for attending today. We'll start this presentation discussing stable isotopes and the capabilities of our stable isotope lab at EMSL, and provide some examples of projects from the lab, like Linda had mentioned. So we see spatial organization throughout nature, and this represents various types of reactions and interactions. To consider spatial interactions, we need to apply appropriate sampling and analytical techniques. One of these methods is stable isotope analysis, and it can help evaluate reactions and interactions. So in our lab, we mainly look at carbon, nitrogen, oxygen, and hydrogen. When we consider the ratio of heavy to light isotopes for an element, where the heavy isotope is rarer than the light isotope. Heavy isotopes behave almost the same as the lighter isotopes, where there are very small shifts in the isotopic composition related to different processes. And so we use the instruments in our lab that are very sensitive to look at these very small differences. We also use these instruments to look at labeled systems where the heavy isotope is added as a tracer. Here we're looking at the natural variation of delta 13C of different materials. So delta 13C represents the 13C to 12C ratio. There are different ranges for different categories. For example, C3 plants look different from C4 plants. And these both have a different range in delta C values than methanotrophic bacteria and fossil fuels. So when we add a stable isotope tracer, we will see a more positive delta 13C value. And this will fall off of this plot. So because the instruments we have are designed for natural systems, it's important to work with us to understand how much labeling is needed for specific experiments when using labeled materials. So stable isotope analyses have been used in many different fields, including topic areas, including biogeochemical processing, carbon sequestration, and various types of forensics. Analyses can provide information on, for example, flux quantification, metabolic processes, information about specific organisms involved. For example, what was a microbe eating, and spatial localization of processes. The instruments we use in our lab includes spectroscopy and spectrometry. In terms of spectroscopy, on the left is our water isotope analyzer. We use this specifically to look at the oxygen and hydrogen isotopes in waters. On the right is an example of an isotope ratio mass spectrometer, or an IRMS, of which we have 4 in our lab. So the basics of IRMS are as follows. So we add a purified gas sample like CO2 to the inlet system. The gas is ionized and races through a vacuum flight tube, passing through a large magnet. Along this path, molecules with heavier isotopes are separated from those with lighter isotopes. For example, 13 CO2 versus 12 CO2. These separated molecules are then collected in Faraday cups, marked by the collector, and measured as voltages. These are related to different masses. Then we use these data to calculate our heavy to light isotope ratios, and they're presented as in the delta notation in per mil values. So in this example, I was talking about CO2, but that isn't the only isotope system we consider. So while we're using a purified gas for the analysis, our samples can start as solids, liquids, and gases, either purified or mixed. And so these samples are purified through chemical, thermal, and physical, or chemical and thermal reactions, and also through physical separation before going into our instrument. So the majority of the work we do in our lab is called continuous flow IRMS. And this is where the samples are automatically processed before entering the instrument. So we can look at bulk and also compound specific samples from mixed samples. When we're looking at bulk samples, we look at organic and inorganic materials in addition to waters. We use the elemental analyzer and high temperature conversion elemental analyzer attached to an IRMS. And we primarily use these devices with the IRMS to look at carbon, nitrogen, hydrogen, and oxygen of samples. So this study utilizes EA, or elemental analyzer IRMS. Using stable isotope labeling, carbon isotopes in the soil system are analyzed to evaluate the impact of perturbation on a system. For example, drought. So 13C labeled CO2 is added to an environmental chamber, and then samples are taken from the rhizosphere, their surrounding soil, and the bulk soil. And what we see here is that the rhizosphere is enriched in 13C relative to the loose and bulk soil. And this helps us quantify photosynthetic flux. So in the previous slide, we saw spatial differences in delta 13C across the rhizosphere and soil. Here we analyzed poplar tissues, so leaves, stems, and roots, after treating the poplar tissues with auxin, which is a root growth promoting hormone. We then labeled the plants with 13CO2 and analyzed all of the tissues to see how carbon allocation within the plant tissues occurs. The root tissues with the highest lateral root growth had the highest 13C. So this highlights that higher carbon allocation to that region of the root occurs, especially under auxin treatment. By performing a separation prior to analysis on our samples, hyphenated techniques like gas chromatography, IRMS, and liquid chromatography, IRMS, can provide isotopic analyses for complex mixtures, thereby providing additional information and higher discriminatory power. You can learn more about a sample than just getting one measurement from the bulk. So in this example, 13C labeled bicarbonate was used as a tracer to evaluate autotrophic and heterotrophic substrate incorporation into microbe-produced amino acids. In this plot, different amino acids are plotted on the x-axis, and delta 13C is plotted on the y-axis. And I know the text is kind of small there. But the amino acids with the most positive delta 13C values, these are the highest bars, are incorporating more of the 13C labeled bicarbonate tracer. So far we mainly discussed carbon isotopes of organic material, but we can also look into other isotope systems of organic material like oxygen and nitrogen. We can also analyze inorganic materials. For example, the oxygen and carbon isotopes in carbonate systems. We analyze those using what is called the gas bench device. And so with this device, carbonate is digested in phosphoric acid, and we sample the headspace for CO2. This system also allows other types of analyses and isotope systems. Now Vimal is going to take over and he'll talk about our laser ablation IRMS system that we have in the lab, and that was made in-house.
Okay. Hi, everyone. Yeah, so Sophie was talking about how isotope analysis can be done with samples that were collected in different spatial spots, like for example, different tissue types or different soil depths. I'll be talking about a technique where you can look at spatial composition within a sample and in the micrometer range. For example, this instrument, laser ablation IRMS, is a EMSL custom build technology where we can look at spatial composition in 50 micrometer spatial spots. Right now, we are using this instrument for carbon-13 analysis. And the way it works is the sample, which is a soil core harvested from a rhizo box, is placed in this ablation chamber. And then the samples are ablated with laser spots that captures 50 micrometer region. And then the ablated samples are carried to a combustion reactor where it is combusted to generate this inorganic gases like CO2, which is then trapped in a cryotrap and then analyzed using IRMS. To explain more about this technique, we conducted a project recently where we looked at maize genotypes, where we had a control maize and then a mutant maize which had an enhanced stomatal opening. So the hypothesis here is the mutant maize with enhanced stomatal opening is going to capture more CO2, and will it transfer that carbon to the rhizosphere, and will there be a difference between these genotypes in the rhizosphere region? For those who don't know, rhizosphere is a region of soil that is contacting the root and has a high metabolic and microbial activity. So in this experimental setup, we had two genotypes, two treatments like well-watered and drought stress. And then we grow the plants in these rhizo boxes. And then there were some non-invasive sample collection like MALDI MSI, which Dushan will talk later. So with regard to LA IRMS, these rhizo boxes were then put back inside this plexiglass chamber, and then we injected 13 CO2 for a period of 2 days to pulse label the plants with 13 CO2. So once the labeling has been done, we froze the entire rhizobox, and then we used a coring device to sample this region containing a root and then its immediate rhizosphere soil and bulk soil. So once we harvested the core, they were carefully compressed and then lyophilized to remove water. And then the right side, you see the lyophilized soil core. containing the root and immediate soil surrounding it. So overall, we harvested about 24 cores, and these 24 cores comprise biological replicates, treatments, genotypes, everything. So once we harvested the core, we proceeded with LA-IRMS workflow. So you see two figures on the left side. This is how the soil core looks before we started the laser ablation. And on the right side, you see various lines of black circles. These are the ablation spots where we started ablating regions close to the root. And then we also collected independently on the roots as well. So moving away from the root, each spot represents 50 micrometer sample size that we harvested for isotopic composition analysis. And when we go far away from the roots, we can adjust the spacing where we looked at intervals of 100 micrometer spots. So all we were interested is to see between the genotypes, are we seeing a difference in isotopic composition when we move far away from the roots? And this is an example plot showing how the isotopic composition looks, starting with the root, which had really high delta 13C values. And when we go far away from the roots, when the distance increases, where the distance is plotted on the x-axis, the isotopic value was immediately higher in the soil close to the roots, and then it drops when we go far away from the roots. Overall, we captured 1,550 micrometer region away from the soil, and then a typical rhizosphere soil is around 1,000 micrometer. And it can vary depending on the plant species, environmental conditions, etc. So the same graph is plotted as a figure on the right side, where you can see on the roots, this red circle represents a higher 13C value. And when we go away from the root, the value drops to a negative value, which is like a lower 13C value. That means that there is no 13C that is transported from the fixed carbon from leaves to the root. So when we looked at the genotypes and the treatments from the study, clearly the drought-stressed genotypes exudated a higher amount of 13C, and the values were really high close to the root, which is like 50 micrometer away from the root. And when we go far away from the root, the value drops, and the treatments were not separatable when we go far away from the root. So that means the rhizosphere soil, in rhizosphere soil, the treatment had really a strong effect in separating from the well water condition. So overall, with the techniques that me and Sophie outlined, we can look at samples that are different soils, different tissue types, microbial samples, volatile samples by using the different IRMS capabilities we have. And the samples can arise from any of these science areas that is listed on the left side. And then we also have stable isotope labeling procedure in plant lab at EMSL, where we can conduct your experiment and then label and then generate samples for our IRMS lab. You can conduct these experiments in your greenhouse or field or mesocosms, and you can send the samples directly to us as well. There is an upcoming call that Linda mentioned, and you can reach out to us if you want to include IRMS capability as part of the proposal, and we are happy to go over your proposal and help figure it out.
Another analysis capability at EMSL associated with stable isotope labeling or stable isotope probing is analysis by nanosims. And as the name suggests, nanosims works at a much reduced scale, which is many orders of magnitude lower than something like the IRMS that Vimal and Sophie have just talked about. However, nanosims is also a great companion method to the likes of IRMS and also some of the other mass spectrometry techniques and analyses available at EMSL, and even the omics capabilities on offer at this facility as well. And what nanosims does allow is the exploration at the cellular and the subcellular scale and offers insight to spatial organization within samples. Next slide, please, Heather. So I'll start with a really brief description of how nanosims works. And in its simplest explanation, it's a destructive surface analysis that counts ejected ions from a solid sample. So nanosims can measure both cations and anions, and including mono and small polyatomic ions, and it works across the entire periodic table. The nanosims instrumentation has really high mass resolving power, meaning that it can measure isotopes and closely related or or ions with very close masses, so we can separate those two out, and that's a unique feature of nanosims. So you can see on the image on the screen here, a very simple schematic of what the nanosims is all about. We have a primary ion source which bombards our sample surface, ejects secondary ions, which are collected and sorted based on mass charge. And these ions are basically counted by movable detectors. So the nanosims here is equipped with 7 detectors. And this is the point of difference between other analogous techniques like time of flight systems that you probably hear so much about. We actually have to tell the machine what ions we're interested in collecting. So we get the output like on the bottom of that diagram where we're collecting 12 carbon, 13 carbon, the cyanide, sulfur, chlorine, aluminum, etc. And we can change those around depending on the needs of the experimentation. But we're interested in isotopes today, so if we focus on the 13 carbon and 12 carbon images that are simultaneously being collected, what we can do is overlay those and generate a ratio image to work out the concentration of 13 carbon. And in this example, we see some yeast cells, and we can create an image where we can look at individual yeast cells, extract the data from all those pixels associated with that cell and generate a value. So in this instance on screen, 2.1 atom percent, 13 carbon with a little bit of error associated. So you can see the potential there. So nanosim is also a very high resolution method, and it has a beam of about 50 to 100 nanometers in diameter. And what this means is that we can raster that beam across the surface and combine those data points to generate images like you can see on the images on the side of the screen here. So next slide. So the next few slides, I'll just give a couple of very simple examples of how nanosims is used. Firstly, nanosims can and does get used on quite a lot of natural and chemically unaltered samples, but today we're interested in stable isotope probing, so I'll go to that space. It's in this arena that nanosims really shines because we don't have the accuracy and precision of some of these more bulky methods. We rely on isotope probing and labeling to generate really great data. So the example on screen here it's the simplest experiment I could think of, consisting of a pure bacterial culture grown in flask. And those cultures have been incubated with a 15 nitrogen, which is the rare isotope of nitrogen labeled ammonia, incubated for a while to see if those cells could accumulate or utilize that 15 nitrogen source. And you can see on the lower left image here, the lovely colored dots, these are all representative of individual bacteria. And using the color scale in the middle there, we can see 15N accumulation ranging from negligible, none basically, all the way through to 65% accumulation. So we have a range of different cells there. Some are blue, so low accumulation or no accumulation, all the way through to yellow, heavily enriched with the 15 nitrogen. And this is in contrast to the negative control, always important in experiments, where just the natural abundance ammonia is added, and we see a blank image. I can assure you there are cells there. The SEM of the same spot shows that there are cells, but there's just nothing above the natural abundance limits. And this is a great result for us. So next slide, please. So this next example is a slightly more complicated system, and it's a multi-organism system. And in this instance, it's a coral system with its associated symbiotic algae. And in this experiment, 15 nitrogen labeled nitrate was added. And the top example here is the coral grown in optimum conditions. And you can see the two large features there, the algal cells are able to accumulate that 15 nitrogen that was added and grow normally. The lower example here shows the same system, but in a heat-stressed environment. And this shows that the algal cells are unable to utilize that nitrate. That's evidenced by the lower right image here. The algae is all blue, representative of natural abundance levels of 15 nitrogen. What you can also see in this example is that nanosims allows us to probe subcellular structures. So the algae on the top right here clearly show differential accumulation patterns, and they correlate well with the ultrastructure of the sample. So the different organelles involved, nucleus, mitochondria, and whatnot. You can also see in this example that we're starting to see transfer of that 15 nitrogen from the algal cells and into the coral tissue like we'd expect. And that's evidenced by this sort of lighter green here around those cells, and somewhere around that, the 2% 15 nitrogen accumulation. Next slide. So this third example is another iteration, and this time we're using multiple isotope labels. So we can add a few different isotope labels at a time. In this instance, we've used 15 nitrogen gas and 13 carbon labeled carbon dioxide at the same time. And this system was a consortium of a cyanobacteria and the associated bacteria. And what we can see here is that we have differential labeling of these cells of the two different isotopes. So for example, we see the vegetative cells of the cyanobacteria, the bead-like structures, have accumulated both the 15 nitrogen and the 13 carbon, whereas the filamentous structures here have predominantly only accumulated the 13 carbon with no or negligible 15 nitrogen uptake. Finally, we have a heterocyst structure in the middle here, in the middle of those beady structures, and this structure actually didn't accumulate either of the 13 carbon or 15 nitrogen. So next slide, Heather. So the final example is a much more complex system. In this system, we're dealing with a small grass species, a plant that's been inoculated with a bacteria, the Aspergillium. And the idea here is that we used a dual labeled acetoamide with 13 carbon and 15 nitrogen, and that was applied to the rooting zone of the plant. And the idea was that the bacteria, that Aspergillium that was associated with the plant, would convert that particular chemical into a useful form that the plant could also uptake. In this case, it's an auxin. So we can see on the right of screen here, this large image, beautiful colored image. We see the bacteria at the top, pink, that shows that there's high accumulation of that 15 nitrogen labeled compound. And we also see that the plant tissue is pretty well labeled as well. So we're looking at 7 atom% of 15 nitrogen accumulation. And this is indicative of transfer of that converted 15 nitrogen compound into something that the plant could use, which is another interesting use of that. So next slide, please. So if you think nanosims is the way to go for your particular sample sets, you're probably interested in what a nanosim sample looks like. And we have 3 golden rules. And if we can say yes to all of these, the samples, there's a good chance that we can analyze it. The first one is that the sample needs to be flat. We're dealing with a surface analysis technique, and so we need a flat surface. We can do that by generating sections, which is very handy for biological systems. We can also create polished surfaces. The geology and mineralogical samples are quite good for that. Furthermore, we could also use small particles. So you saw in the previous examples, we were just using bacteria, which can just simply be aliquoted onto a stub of some description, and we can dry those down and analyze them directly without any fancy sample preparation. That works quite well. The second, probably most important, is that the sample needs to be dry. This is obviously a problem for biological stuff because there's a ton of water in there. And the reason for this is that the instrument works under ultra high vacuum. So dry sample allows us to achieve that. Wet samples, obviously, we're going to have all kinds of problems there. And finally, the samples need to be conductive. This is the lesser of the 3 evils, and it's usually reasonably easy to overcome. And that might be just by coating the sample with a couple of nanometers of a metal, like gold, iridium, platinum. It doesn't really matter, as long as it's not something you're interested in analyzing. The instrument's also fitted with an electron flood gun that can compensate for charge. So for insulating samples, we can use that to overcome those problems. So if you can answer yes to all these questions, and the size of your samples is compatible with these sort of sample holders examples that I've got on screen pictured, there's a good chance that we can do something for you with the nanosims instrumentation. So just the next slide, please, Heather. So as a final slide, a few tips and tricks for what a nanosims type experiment might look like. Number one is to make sure you've got well-defined aims, hypotheses, and expectations. This is a very targeted method because we have to we're telling the instrument what we want it to analyze. So we need to have great hypotheses in place and understand specifically what we're looking for in terms of that. And also know your end game. What's the data that you particularly want to get out of this? Are we looking for statistically viable numbers for cell counts? How many cells took up a particular isotope, or what competition factors, et cetera, et cetera? Are you just interested in getting some nice images to show a particular pathway of interest? So this very simplified diagram shows all the aspects of the experiment that we can interchange and that are important. you're ranging from the isotope tracer that we're adding, because we can add any isotope tracer using this instrumentation. The system, of course, that's up to the user. What particular system are they interested in? The treatment, what are we going to do to that? Obviously associated with the scientific question. The time of incubation is probably an overlooked issue with these samples. How long are we going to leave that isotope there? Because it's going to transform, it's going to move, it's going to change. So that's a very important, and it may be that the experiment is actually to test this particular parameter. Sample preparation, as I've alluded to, is quite tricky with these, or can be quite tricky with these particular analysis, and it's something that really needs to be considered for every project individually. Also like to keep in mind that NanoSims is really just a fancy mapping tool to show us where these isotopes are accumulating. So we need to think about the companion techniques or the correlative techniques that we're going to work with, whether the omics or other mass spec-based methods. And it's important to keep that in mind in terms of, are we going to be analysing these samples for both methods and questions like that? And these are the questions that we here at P&L can help you answer, and we'll spend a lot of time with you to sort of elucidate before we embark on some sort of instrument analysis. So I'll hand it over to Dushan now, and he can explain some other exciting capabilities.
Perfect. So yeah, thanks Sophie, Vimal, and Jeremy. So, and thank you for being here. So now we will go in a little bit different mass spectrometry imaging capability. NanoSims is one great capability that we saw for like tracing isotopes and elements. But here I want to focus more on the basically untargeted molecular imaging, and this will be on the roots and the rhizosphere. But we can talk later about that it can be also like different other environmental systems. So this technique that I will show you today, called MALDI, mass spectrometry imaging, and Jeremy already actually what you will learn today is just basics about mass spectrometry imaging, and Jeremy already kind of hint about that, so I will not spend too much time here. I will mostly focus on MALDI mass spectrometry imaging and how we actually use MALDI mass spectrometry imaging for spatial metabolomics at EMSL and for the EMSL users, focusing on 3 different examples. So in imaging of intercellular metabolites, so cell type specific metabolomics, let's say in the root tissues, for example, and then how we can also image and track extracellular metabolites or root exudates within, for example, synthetic soil habitats like rhizochips are, or within native soil environments inside the rhizoboxes, for example, that Vimal was mentioning in first part of the webinar, using technique called rhizomap. So just briefly, that for mass spectrometry imaging is basically spatial probing of sample surface chemistry, and then we have like basically specific probe. We saw in the NanoSims that we use a primary ion beam, but this probe can be, for example, laser in case of the MALDI, or some liquid droplets. You can also think about that where we basically probe a particular area in the sample, and then we basically transfer those molecules or ions into the mass spec, where basically mass spec present actually kind of reflect molecular composition of that particular area that we probe. So in that way, like mass spec imaging, you can see as a really tool that can reveal you spatial distribution of molecules within biological samples, especially in the MALDI mass spectrometry imaging, in almost untargeted way, where we can basically visualize all the way from small molecules to the small proteins. Today, I will focus mostly on the metabolites. And then depending how well we can focus our probe, we saw in the NanoSims that we can go in the nanometer scale range, so in the subcellular range. But other techniques, like you will see in the MALDI, we are limited, let's say, on the cell scale, like 100 or 5 micrometers to 50 micrometer cell range. So we can basically, depending on what capability you want to choose at EMSL, and EMSL is really equipped with a lot of tools that can enable analysis of environmental processes at different scale, you can pick what tool is really good for answering your question. So MALDI stands for matrix assist laser desorption ionization. So in MALDI, the most important component is actually matrix. So it is some, let's say, small aromatic acid or base that we add in our sample that basically boosts ionization of our analytes. And it can also kind of serve as tool that can choose what kind of molecules we will analyze. So depending of what MALDI matrix we apply, we can, for example, analyze amino acid components of TCA cycles, phytohormones, phenolics, alkaloids, sugars, biogenic amines, lipids, but very often just by using one MALDI matrix, we can cover most of those molecular classes. Common lateral resolution that is largely depend on the focusing of our laser is 5 microns to 20 microns. So that is something that we do on a routinely base in EMSL. So we can go like you, and you will see on the examples on really like near single cell level. And in EMSL, we offer two different multi-mass spectrometry imaging capabilities for spatial metabolomics. We have also another one for even for spatial proteomics, but mostly like those are MALDI FTICR, which is actually, I will not go too much in the detail, but it provides you really ultra high mass accuracy of measurement. So we are really accurate in the molecular formula of the molecules that we image. And then we offer also MALDI timsTOF that is limited a little bit in the accuracy and mass resolution, resolving power, but it has really high spatial resolution and high throughput. And in EMSL, usually for our projects, we analyze samples on both instruments so that we can get idea to actually information about really high spatial information, but also accurate information about molecules that we image. So first I will go over how we do intracellular metabolic imaging. Basically, you can see actually on the previous slide what is usually workflow that we kind of harvest the roots, embed them in cryoblocks, cryosection them, do microscopy of them, and then do all this MALDI matrix coating, MALDI analysis and data analysis. So you can see here that depending of what type of MALDI matrix use or what type of workflow we use, we have recently developed on tissue chemical derivatization workflow that can basically also reveal some elusive compound and some compounds that are really in the low abundance, like for example phytohormones, but also some other aliphatic acids and aldehydes. We can basically tune what molecules we will see, and you can see that in this graph here that depending of what matrix we use, we have different metabolic coverage and different primary and secondary metabolites we can detect and image in the tissues. This is just example how by using one tissue chemical derivatization, for example, we can image almost entire TCA cycle, but also phytohormones, for example, in the poplar root cross sections. So usually we deal either with cross sections or with longitudinal sections of the roots, and then, as I said, like we capture microscopic images so that we can afterwards overlay our MALDI images with microscope images and get information about cell specificity. So this is example how, for example, methyl glyoxal, one stress reporter, we were able to visualize in the vascular cells of the poplar root, and then you can see also that that, for example, when we analyze the lateral or not lateral but longitudinal section of the roots, we are able to see that, for example, in the drought conditions, we have accumulation of the aspartic acid in the tips of the roots. But we can also go in analyzing not only intercellular processes, we can analyze what is going on with the root exudates in extracellular space. So for that, we offer users to use our synthetic soil habitats that basically can really in controlled manner mimic what is going on in the soil. So those are calling rhizochip, and they're made compatible with multi-mass spectrometry imaging. And then basically by analyzing those rhizochips, you can have idea. Basically, you can cover really like a lot of different, like roughly like around the 400 to 500 molecules we can image from those rhizochips. But actually, you can see what type of molecules are released in what type of the roots, but also how far from the roots going in those synthetic soil pores. We also have capability that can analyze it in the native soil, basically using the rhizoboxes that Vimal was mentioning. So in this case, basically we use a different type of the membranes where we imprint molecules from root soil interface or from the soil to the membrane, and then basically use those then use multi-mass spectrometry imaging to image those membranes and to reveal molecules that are imprinted in situ on the membranes. And as a result, you have like data that looks like this. Basically, that you can visualize and track root exudates through the space, so to the depth of the rhizosphere. But also because this is non-destructive techniques, you can just place the membrane from time to time in the different growth stage of the plants. You can actually track those root exudation in time. So with this, I would like to finish and to tell you that all those capabilities are available for you. And if you have any questions or you want to, apply for the FICUS project, yeah, please let me know. So yeah, I think that's it.
Fantastic. Thank you, everyone. We are going to move on to the Q&A because we have received an abundance of questions from the audience, which is very exciting. We love that you are so active in the questions, and we can't wait to get to them. So without further ado, I am going to rotate. I won't necessarily go in order of the way that we receive the questions, just so that everyone has a chance to answer some questions. So we are going to start though with Sophie first. Sophie, we received a question from Anonymous. Can you explain how you have a negative delta 13 carbon percentage?
Yeah, so something I neglected to really lay out was how we get these delta 13C values or delta values in general. And we're basically measuring, or it's basically a calculation of a ratio of ratios where you're looking at 13C to 12C of an unknown over 13C to 12C of a standard. And when you have a negative delta 13C value, that really means that there's less 13C in that unknown than there is in a standard that we use. So that's what we mean by negative values.
Great. Okay, next one for Vimal. Can you handle pathogens with the LA IRMS?
Okay, yeah, we have the plant lab in EMSL is BSL1, where we mainly look at beneficial strains, but we have other facilities with BSL2 access, so we can handle plant pathogens there. Yeah.
Fantastic. Okay. So next for Jeremy, Hamidreza is asking, is there a minimum or recommended sample volume for the analysis using nanosims?
That's a tough question to answer. I guess, how do I answer this? Minimum value. So we're looking at solid samples in these particular instances. So a lot of the times it's plant tissues, for example, or microbes onto a cell. So we can actually analyze a single microbe. Keep in mind that the resolution of nanosims is very small, so our beam is very small, so 50 to 100 nanometers. So that's the minimum analysis size. In theory, though, we want to actually combine quite a few of those analysis points to generate more meaningful data.
Okay. Okay. Next for Dushan, we have what are some sources of isotopically labeled compounds? David is asking you for, and I believe this is for Molly.
Let me see. Is it
What
like
are
for,
some sources of isotopically labeled compounds?
is it a question for me?
It came in during your presentation, so it might be for Jeremy or
I
for
think
you.
it's more for Jeremy. I mean,
Okay.
yeah.
Yeah, probably more for me. There's plenty of sources out there, ranging from somewhere like Sigma, Cambridge Isotope Labs in the US is quite good. Just a quick Google search will find many, many options out there, even to the point of looking for labeled plant biomass and things like that we can add into experiments. So you could also make some yourself, make more complex compounds or tissues that you might be interested in.
Right. Okay. So another one for you, Dushan. Let's see.
Yeah,
How
I saw. Yeah,
how
yeah.
how quantitative is MSI, and how confident are you in that quantification?
Yeah, so MSI, I mean, we don't do absolute quantification here. So that's one thing, unless like we have like really targeted compounds. So then, like we can do it. But like what we provide here is mostly like relative quantification and relative abundance. So we are not doing, unless it's like really like we select 2-3 compounds, then we can do like absolute quantification. But in this case, because like multi-process is not even like your signal intensity like between two compounds, different compounds, doesn't mean if one signal intensity is higher than from another that compound is more abundant. It just depend of the ionization properties. So, but yeah, to make it short, like we don't do absolute quantification here. Everything is based on the relative quantification.
Okay. Vimal, how did you prepare your soils, or how do you prepare soil samples? Do they need to be fresh, frozen, dried?
So we use regarding the kind of soil, if you are interested shipping your soil, we can have them here. Because if you're looking at specific soil with specific microbiome composition, you can ship the soil to us. We can set up the experiment with your soil. But for the LA IRMS, once the plant growth and the labeling has been achieved, then the entire soil has to be frozen because the drill bit operates in liquid nitrogen condition. When we drill the soil, it shouldn't collapse. So we drill the soil and then we compress it to make this soil pucks, and then lyophilize it, and then stage it on the instrument. So pretty much there'll be a set of soil processing that'll be happening after we finish the plant growth and labeling.
Okay. Let's see. For Sophie, and Vimal, so Karina has asked or wanted to give feedback. So this is really neat tech and your approaches. Could this rhizosphere carbon tracking be applied to the mycorrhizosphere or the hydrosphere if fungal establishment is part of a rhizobox? For instance, tracing carbon signature and nitrogen signatures from root to hyphae and vice versa. Maybe it could be a great nanosims pairing, but that ablation and root spacing is really great. So anyway, do either of you want to take that?
We
Great. I
can
mean,
go ahead, Vimal.
yeah, sorry. Yeah, so the resolution is 50 micrometer. So if we can be able to, that is the size of a plant cell that I know. So when it comes to differentiating root and fungi, probably we can be able to do that. We haven't done that yet. So that's kind of my answer.
I'm wondering if it's if you kind of have bags that contain some of the systems, so the mycorrhizosphere, for example, but we can in general look at more than one isotopic system at once. So we could look at carbon signature and nitrogen signature. But with laser ablation, that is something we've been wanting to do, is to get nitrogen signatures using the LA-IRMS system.
Okay. We have 5 minutes left, and we have a lot of questions still that have come in. So, Jeremy, another one from Morgan. Are there specific filters that cells can be collected on that are more compatible with nanosims? Do filters have to be sputter coated, similar to prep for SEM?
Yeah, that's a typical technique when we're dealing with filtered microbial cells and whatnot, is just to use usually a glass slide, or a GFP slide or something like that. We're not too fussy. As long as it's conductive and we can make it conductive. So yeah, we often will pre-coat them with gold, just a really fine layer of gold to help the process out. So it sounds like you already knew the answer to that one.
Great. Okay, for Dushan, so Wesley is asking, does the MALDI laser probe preferentially ionize certain compounds? Can the laser probe be adjusted to target specific compound classes?
Yeah, so as I said, so it's pretty so basically, yeah, so laser that we use is like a UV laser, right? So the technique is biased a little bit toward aromatic compounds and to compounds that kind of absorbed in the UV range. But that doesn't mean that, you know, like we cannot analyze non-aromatic stuff. Actually, like we can, and it is reason why we add like even our MALDI matrix. It is aromatic based, like actually aromatic acid or base that help in ionization of other compounds. So basically, it is not that, as I said, it's really mostly like choosing of the MALDI matrix will kind of govern what type of molecules you will see. But we have like some, maybe like 2-3 matrices that we use here that kind of, if we use those 3 matrices, we can cover basically a lot of different compounds. One thing that is like really kind of hard to detect, what I've experienced, is like steroid compounds. But all other stuff, I mean, obviously, like if they're in the low abundance, like phytohormones, it is reason why we needed to do like to boost ionization efficiency by adding like different derivatization compounds. But it's not, you know, it's like really untargeted approach. Basically, in one experiment, you can detect anywhere for the components of TCA cycles, even like lipids in the same run. So it's not that laser, you know, will dictate what kind of molecules will be mostly MALDI matrix that we choose, or in that sense. I don't know if that really answers your question, but yeah, it's like really broad range of molecules that we can detect
Okay.
in single experiment.
We have 2 minutes left, and we have so many questions. So I just want to let the audience know, we obviously are not going to be able to get to everyone's questions, but that's okay, because since you registered, we have your email address. We can have our folks here will send you an email back with the answer to your questions. So just know that it'll get answered even if we don't speak about it here. And I think we have room or time for maybe 1, possibly 2 questions, depending on how quick you can answer them. Dushan, I'm going to have one more for you. Actually, no, there was a couple questions about the ficus proposal. So
Yeah.
I think let's start with that one. I got to find where that one was. There was a question. Apologies. Okay, these are really amazing so Karina, these are really amazing capabilities. It seems that ficus could propose to incorporate several of these capacities or capabilities for an EcoFab or Rhizobox project. But how would that work with contacting EMSL scientists? Do I pick one and contact all once the pre-proposal is written to get feedback? How would you suggest that she go about this?
You ask me or like anyone? Yeah,
Anybody.
I mean, usually like users like approach, yeah, to the expert, like, yeah, for each of these capabilities on the EMSL site, you have like contact persons, right? So, but anyway, like you can contact any of us and we can guide you towards like Vimal, Jeremy, or some other technique if you want to use, right? And then we together, let's say, write the proposal, right? So especially like, I mean, for letter of intent, you can contact one of us or, and then we can help you about that. But then like when we draft like full proposal, like Vimal, Jeremy, Sophie, all will give like their expertise, like how for sample preparation to like data that you can expect. So yeah, I mean, talking with us is like really like important for successful proposal.
Yeah. So Karina, go ahead. You can email one of the folks
Yeah.
here and then they can help get you to the right person or bring in more folks.
Yeah.
And it's 1 o'clock, so our webinar is uh, finished, but just again wanted to thank everyone for your questions. We will have an answer back to you via email, so thank you for that. And also to let you, if anybody's still on here and is listening, there is no silly questions. I'm seeing this a lot in the questions, like, sorry if this is a dumb question. Don't worry, nothing's a dumb question here. So much of this is complicated. So, yeah, we're here to help you figure it all out. So, hope everyone has a great day, and we will see you at the next webinar.